Juxtaglomerular apparatus is a complex, specialized structure found in the region where the tubule lies in contact with its own glomerulus, forming a specialized cells of varying functions. Juxtaglomerular apparatus plays an important role in feedback mechanism called tubuloglomerular feedback mechanism, which regulates glomerular filtration rate and blood flow through glomerular capillaries. These specialized cells includes macula densa, Lassie cells, or they're also called as extraglomerular mesangial cells, and juxtaglomerular cells. Macula densa. Macula densa are tightly packed specialized group of epithelial cells in the distal tubules that come in contact with afferent and efferent arteriole of its own glomerulus. These cells are or can detect sodium load in filtrate and can regulate secretion of renin. It is likely that macula densa mediated renin secretion helps to maintain systemic arterial blood pressure under conditions of reduced vascular volume. Lassie cells or extraglomerular mesangial cells. Lassie cells are mesangial cells located outside the glomerulus, that is, they are found in the triangular area between the afferent arterioles, efferent arterioles, and macula densa. They are agranular cells. They produce renin in small amounts. These cells are contractile and they can play a role in regulation of GFR by regulating blood flow through the glomerular capillaries by altering the capillary surface area. Juxtaglomerular cells. They are modified smooth muscles lining the afferent arterioles near glomeruli. They are granular and can synthesize, store, and release renin. The juxtaglomerular cells are in sympathetic nerve fibers and release their renin content in response to sympathetic discharge. They act as the baroreceptors. They monitor renal perfusion pressure and are stimulated during hypovolemia or decreased renal perfusion pressure. So this is an important diagram showing the juxtaglomerular apparatus, which includes afferent arterioles where the cells are modified as juxtaglomerular cells, showing the distal convoluted tubule, which are the cells modified as a macula densa, which can detect sodium load or chloride load, and we have also the Lassie cells or mesangial cells. Renin angiotensin system. Cells in the afferent arterioles that are sites of synthesis, storage, and release of proteolytic enzyme renin. Some of the stimulants for renin secretion are whenever there is a fall in arterial blood pressure, reduction in the ECF volume, increased sympathetic activity, or decreased load of sodium or chloride in macula densa, prostaglandins can stimulate renin secretion. The renin is produced from the juxtaglomerular apparatus, that is juxtaglomerular cells, and where it is going to convert angiotensinogen into angiotensin 1. Angiotensinogen is produced in the liver, and the converted angiotensin 1 is going to be released into the circulation, where in the lungs, with the help of angiotensin converting enzyme, it is converted into angiotensin 2. And angiotensin 2 is a profound vasoconstrictor, and it can increase sodium and water reabsorption from the renal tubules. And some of it is also going to be converted into angiotensin 3 and 4 as well. Inhibitors of renin secretion. Increased load of sodium and chloride in macula densa, increased afferent arteriolar pressure, angiotensin 2 via negative feedback, and vasopressin can inhibit the renin secretion. Renin alone does not have any physiological function. It functions solely as a proteolytic enzyme. Its substrate is circulating protein angiotensinogen, which is produced in the liver. Angiotensinogen is cleaved by renin to yield decapeptide angiotensin 1. Angiotensin 1 also has no physiological function, and it is further cleaved to octapeptide angiotensin 2 by converting enzyme, that is angiotensin converting enzyme secreted from the lungs. Various peptides Peptidases converts angiotensin 2 into angiotensin 3, which is a heptapeptide, and angiotensin 5, which is a hexapeptide. Some of the actions of angiotensin 2 are: it causes vasoconstriction leading to increase in the arterial blood pressure. It increases the blood pressure directly by increasing the release of norepinephrine from postganglionic sympathetic fibers, where the norepinephrine acts as a vasoconstrictor. It acts directly on the adrenal cortex to release secretion of aldosterone. We know that aldosterone it is a mineralocorticoid. It increases the retention of sodium. It also acts on brain to decrease the response of baroreceptor reflex. The angiotensin 2 increases the water intake by stimulating thirst center, and it also increases the secretion of ADH and ACTH. Angiotensin 3 has a pressor activity and aldosterone stimulating activity. So these are some of the the angiotensin mechanism. Angiotensinogen is released from the liver. It gets converted into angiotensin by renin. In the lung, it gets converted into angiotensin 2. And angiotensin 2 has the profound action on the adrenal cortex, which is going to produce aldosterone. It has direct action on the renal tubules to reabsorb water. And the ADH is mainly going to inhibit the activity of angiotensin 2. Some of the applied aspects related to juxtaglomerular apparatus are: angiotensin converting enzyme inhibitors are drugs used in the treatment of hypertension and congestive heart failure. They inhibit the formation of angiotensin 2 and thus decrease